This is part 5 of the cell lecture. We left off with the Golgi apparatus. We are learning about cell organelles. The Golgi apparatus is made of flattened stacks, kind of like, to me, like they're kind of like pita bread, stacked on top of each other. What the Golgi does is modifies and packages the products that come from the smooth and the rough ER. And the Golgi modifies and packages those secretions for release either into the cell or outside the cell. The Golgi helps renew or modify the plasma membrane. As you can see, the Golgi will pinch off at the ends and form vesicles, and those vesicles can fuse and join with the plasma membrane and increase the size of the plasma membrane. The Golgi apparatus is responsible for producing lysosomes, because lysosomes are vesicles that contain digestive enzymes inside of them. So the enzymes are produced by the rough ER, and then they're transported to the Golgi apparatus, and the Golgi apparatus packages them into a lysosome. So lysosomes, again, are defined as vesicles filled with digestive enzymes. They function in removing damaged organelles or pathogens, and also just digestion in general. So they function in cleanup and recycling old and damaged cells or damaged organelles, defense against disease, because white blood cells have lysosomes that will fuse with pathogens and break them down. And then autolysis is when cells are damaged, and sometimes autolysis is a little bit different from apoptosis. Apoptosis is programmed into the DNA. Autolysis is when the cell is damaged, the lysosome will actually break down the cell and digest it. It digests itself, so it's called autolysis. So apoptosis is a form of autolysis. Peroxisomes are similar to lysosomes, but they are primarily involved in breaking down fatty acids. Mitochondria are responsible for producing almost all the ATP required by the cell through the process of cellular respiration. The first step of cellular respiration is breaking down glucose. It's called glycolysis. And the remaining steps of cellular respiration or aerobic metabolism occur inside the mitochondria. Glycolysis occurs in the cytosol, in the fluid. Shows you the structure of the mitochondria. It has an outer membrane and a folded inner membrane, and the folds on the inner membrane are called cristae. And remember, anytime when we're studying living things, when we see folds, like the folds of the plasma membrane that form microvilli, foldings increase surface area. So the mitochondria can produce much more ATP with the inner membrane being folded. And I'm going to skip these checkpoint questions because we're already on part 6, I believe, or part 5, and that's enough. So the nucleus is usually the largest structure in the cell. It's the control center. It controls protein synthesis because it contains the DNA. It contains the DNA in a form called chromatin. The nucleus is surrounded by a double membrane called the nuclear envelope. The fluid inside of the nucleus is called the nucleoplasm, and the nucleus does have pores that allow for substances to leave the nucleus. Not the DNA, but other substances like RNA can leave the nucleus. The DNA in a cell that is not actively dividing is called chromatin. Inside the nucleus, you also can have either one nucleolus or you can have many nucleoli, and these structures synthesize ribosomes. They assemble ribosomal RNA into large ribosomal subunits and then connect those subunits together to form ribosomes. So the nucleolus produces ribosomes, and then the ribosomes produce proteins. Chromosomes, humans have 46 chromosomes in all of their cells except their sex cells. The sperm and the egg only contain 23 chromosomes. All the body cells contain 23 pairs of chromosomes, or 46. The chromatin is loosely coiled DNA. When the cell is not dividing, this is how you find the DNA. It's called chromatin. But when the cell is dividing, this chromatin condenses into the chromosomes, the rod-shaped structures that we call chromosomes. And you can kind of see, this is DNA unwound. You can see how it winds itself around a histone protein, and then it's called a nucleosome. And then it coils a little bit more, and we call it chromatin. And then whenever it really, really super coils and condenses, we call it a chromosome. Let's see. The genetic code is the chemical language of the cell, and it comes from the DNA code, of course. Each sequence of 3 nucleotides codes for a single amino acid. So what DNA really does is produces the amino acids that form into proteins. A gene is a piece of DNA that codes for a specific protein. I went over this. Genes are normally tightly coiled with histones, and that keeps them inactive. To activate the gene, an enzyme must break the bonds and remove the histone, revealing a promoter region. And then protein synthesis can occur. And protein synthesis occurs in 2 stages. Transcription occurs in the nucleus, and translation occurs outside of the nucleus in the cytosol. Transcription is when the DNA, the piece of DNA, the gene that's being transcribed, is used to code for messenger RNA, which is a smaller single-stranded instead of double-stranded. It's a smaller molecule that can leave the nucleus and carry the message of the DNA into the cytoplasm or cytosol to where a ribosome can use it to make a protein. Here are the steps of transcription. I don't want to go too much into this. I just want to mention that to form the messenger RNA, you need the enzyme RNA polymerase. What it does is it takes, the DNA code is made of adenine, cytosine, guanine, and thymine, A's, C's, G's, and T's. So, for example, if you had a TAC as as a DNA code, the RNA polymerase would attach an A to pair with that T, and it would attach a U to pair with the A and a G to pair with the C. This would be the code in the messenger RNA that would leave the nucleus, and that triplet code is called a codon, and each triplet code or codon codes for a particular amino acid, or it can code for a stop signal. There are 3 codons that code for a stop signal. That means stop making protein. This shows you transcription. Here's your DNA. The DNA has to unwind, and you only use one side as the template strand. And then the RNA polymerase runs down the DNA, and as it does, it attaches nucleotides to the RNA polymerase. and they form a chain, and then they eventually form into the messenger RNA strand. That's transcription. Translation occurs when the messenger RNA is used to code for the specific amino acids that form the protein. The amino acids are delivered to the messenger RNA, which is attached to the ribosomes. So the amino acids are delivered by transfer RNA. Transfer RNA contains an anticodon that is complementary to the codon in the messenger RNA. So let's say, for example, the messenger RNA has this codon AUG, which codes for the amino acid methionine. There would be a transfer RNA in the cytoplasm or the cytosol that contains the code UAC, and it would also be connected, attached to methionine, and it would move into that spot on the messenger RNA where the AUG is located. This is examples of the genetic code and how they code for amino acids. So if this is your DNA triplet code, AAA, the messenger RNA is going to be UUU, because remember, RNA has U instead of T. And the transfer RNA will be the same as the DNA. The only time the transfer RNA anticodon is different is if the DNA has a T in it. Obviously, since transfer RNA is RNA, it's going to have a uracil in place of the T. So the messenger RNA is going to be complementary to the DNA and to the transfer RNA. And this lists different amino acids: phenylalanine, leucine, cysteine, valine, proline, alanine. Methionine is the one the example I gave you. And then, of course, a stop codon. In this case, there is no transfer RNA for the stop codon because transfer RNA transfers amino acids to the ribosome. And the stop codon does not code for an amino acid. It just it just means stop. So the complementary sequence to the DNA code ATT is UAA, and that's one of these stop codons. And once that is encountered by the RNA polymerase, then translation is over and your protein is complete. There are 3 phases of translation. It starts with an initiation phase, which uses a start codon, and that start codon is AUG, and I've already told you what it stands for. It stands for methionine. Every protein in the entire world, in any organism, begins with the amino acid methionine. All proteins begin with methionine. And then the transfer RNA is going to bring different amino acids. This is your amino acid, and this is the anticodon. Transfer RNA is going to bring the correct amino acid to the correct codon. And the blue structure here is your ribosome. And then then the amino acids will be linked together to form a protein. That's the elongation phase. And then the termination phase is when the stop codon is encountered. And this is showing you a picture of the elongation and termination phases of translation. Okay, now we also need to go over the life cycle of a cell and talk about cell division. Cell division is a form of cellular reproduction. What it does is increases the number of cells, so it can help us to grow larger, and it can help us to repair, like if we get injured, it can help repair our body tissue. Cell division replaces old and damaged cells. We lose our top layer of skin every 2 to 3 weeks, and that top layer is replaced by new cells that are formed from cell division. And cell division can't happen unless the DNA is replicated, because each new cell, here's your old cell has 46 chromosomes. And then when it divides, each new cell has to have the same exact number and sequences as the old cells. So the new cells have 46 chromosomes as well, and you can't have that unless the DNA makes a copy of itself.